I'd like to thank the Pfenex team, Bill, Cassidy, and Christy for inviting SAFC to participate in this conference. As Bill indicated, I've had over 20 years experience working in the contract manufacturing arena and doing tech transfer. What I'd like to do today is to share some of the factors that we found important, maybe share a few anecdotes, and hopefully provide some guidance in terms of what is an appropriate path forward as you move from your innovative work and discovery into the clinical setting and beyond. A little bit, just brief introduction, who is SAFC? We are the commercial arm of Sigma Aldrich. So this is the part of the company that actually supplies into the industrial market. So I know that it may not have the same name recognition, but we are part of the Sigma Aldrich family, and so that's where we come into play. In terms of how are we tied into Pfenex? Well, we have a strategic partnership with Pfenex to use their platform for GMP manufacturing. We have a recently opened GMP fermentation facility in Jerusalem, Israel. It is a beautiful location. And it's a multi-purpose, multi-product facility in that we have the capability to do kind of small molecule manufacturing, kind of secondary metabolite, as well as therapeutic proteins. So we have a variety of reactor sizes, but we have the segregated manufacturing suites so we can handle this variety of product class. In case anyone is wondering, yes, we do have manufacturing facilities worldwide, and why Jerusalem? Other than it's a really great place to visit if you want to see cultural and historical and significant sites. It's where we had our Center of Excellence for Fermentation Science, significant R&D activities there, as well as access to a wide variety of other technology and high-tech areas in that vicinity. So that's why it's in Jerusalem. So what I'd like to do is to share a little bit. We do have a kind of a brief case study, and then in general talk about some of the concepts of tech transfer from the conversations today and based on experience. One of the things as you scale up from your research application into your industrial is that obviously the size of your fermenters increase. So in our facility we have a 19 liter, which is generally our seed fermenter, 100 liter, 400 liter, and the 100, 100 liter, 1,000 liter, and the 4,000 liter fermenter. So that's something, as you scale up, you have to take into consideration where did you start and where do you want to end up at based upon the amount of protein that you need. One of the things that we need to consider are the challenges from a CMO perspective. It's been interesting hearing some of these discussions. There's always an advantage and a disadvantage to being the last speaker. One of the things is we get to see a really wide variety of products. So my life is never dull, it's not routine, there's always something new and different coming down. It's interesting when I hear about these novel technologies because I know at some point, yep, Kevin, I'm going to be one of those CMOs you're going to work with that have never seen your product before but have to figure out how to effectively manufacture it. Have I seen processes where I went from great 50% yield or conjugation efficiency at bench scale, get it into my hands, and what happens? The bottom drops out. Why? That's what we have to figure out. How do we avoid that as we go into tech transfer to make sure it's effective, robust, and suitable from a quality standpoint? I was asked during break, what is the best time to do a tech transfer of your product? What I see is that the development status will vary tremendously among projects. I've done ones where we've been fortunate enough to essentially start in on ground floor with preclinical development, and I've also transferred commercial products where either it could be they had lost a vendor, or it's something that they're trying to resurrect a product, bring it back to life, and we have to demonstrate comparability. In my mind, the ideal time is really when you have done your initial proof of concept work, you know that you have a target molecule that you're interested in, and what you want to do is to bring it into the clinic. The fewer number of handoffs you have between different manufacturing locations, different personnel, generally the higher success rate you'll have. Another thing that's always interesting is timing. When do you need your molecule? How long will it take to get there? Sheila pointed out, you know what? The pathway is relatively long to go from your preclinical studies into your first in human. The hardest thing for us is to get an inquiry, which happens on a fairly routine basis. Hey, I've got wonderful preclinical data. I want to go into the clinic in 3 months. Can you do it? The answer is probably not. We never like to say no, but we will try to temper it with a healthy dose of reality. In reality, even for a well-developed process, you're going to need generally at least 6 months, maybe more, to get all of the required attributes transferred into your facility so you can be comfortable you've got what you need to move forward into the clinic with a high-quality product. And another thing that can't be emphasized enough is your communication flow, and I'll talk a little bit more about that. But when you get down to it as a contract manufacturing organization, one of the primary things that we provide is in our service to you is not only the product, but it is that communication. It's all the documentation that you need to do your filing, that you need to do to characterize your molecule, that you need to have to know whether you're on track or not. So that's something that's a critical component of it. So it's one of those, I think I have, one of the best positions there are. I get to see all of the great innovation, but I get to turn it into your reality so you can go forward in the clinic. The way we view process development is kind of a multi-step approach. And one of the first things that we will want to do is to define what is the product you need. And we will front load the analytical development on that. I understand your position, Kevin, when you talked about, okay, we don't know exactly what our mixture is because we don't have the analytics in place. And this is one of those things that we routinely run into when we are doing a technology transfer, is that we really essentially have to do the process development and the analytical method development concurrently. It's a challenge because you need product to develop your analytical methods, but you don't know if you have the right product until you have your analytical methods in place. So it's a balancing act in terms of getting those put in place. In terms of technology transfer, as I said, a lot of that will be dependent upon where we start with the client. I have to thank Sheila for already kind of covering the quality by design. It used to be that, the concept was, and design of experience sounded like a really kind of nice thing to do. It's the way you should do it, but was often skipped because if you got something to work, you just rammed it forward and figured out, I'll catch up when I get into my phase 3 and commercial product. Guess what? The FDA doesn't agree with that approach anymore, so you gotta think about it when you start. So whereas in my previous years, we would tend to do all of the experimental design work, collecting information around critical parameters, when you were really hitting your phase 3 studies, when you knew you had a real product. What we're doing now is seeing that we at least have to get part of that work done early to support those characterization studies. Ideally, we want to do at least 2 to 3 batches at small scale before we move forward. I hear you when you say we don't have the money to do everything we want to do. And we recognize that's going to be reality for many of the academic labs that have great ideas, or for the biotechs. So that very often we have to do some risk assessment in terms of what is the minimum amount of data that we can collect before we move forward, what's the minimum number of runs we can do before we move forward, and go from there. As I said, one of the very first things we're going to do is to develop the analytical methods. As you can see, they are the cornerstone for a lot of what we do. We're going to use it to identify our process to know if it's suitable. We're going to do it for product testing and release. We're going to use it to characterize. What I don't have up here is all of the cleaning methods. So to date, we've kind of ignored the fact that when I get into manufacturing, I'm going to have to figure out how to ensure my equipment and my facility is clean of any residual product that I made for you before I go into the next manufacturing batch. And that can be challenging at times depending upon what methods are available. For proteins, it's generally more straightforward because you can do a simple TOC for your method of analysis. Protein manufacturing, whether you're doing it on the bench or in the manufacturing facility, the concepts are the same. The scale of equipment and the type of equipment may change because I'm going to need to have more controls around what I use. As we already heard from Sheila, the cell bank creation is going to be one of the first things that you're going to do and put a lot of thought into. Do you know what you have? For a product produced by fermentation, either mammalian cell culture or microbial fermentation, having a well-characterized cell bank is the key to having a consistent product. So that is one of the things that will be essential, is to ensure that you have a high-quality master cell bank as you move forward into your GMP manufacturing. In terms of the optimization as we go through process development, there are the different steps where you want to see how do you get your best productivity and go through that. All of that's fairly standard, but there are some things that you may not think would get you, but they do. Have you ever thought about the quality of water that you use? For more than one fermentation facility, when we've had conversations, you would always think that higher the purity, the better you are. Guess what? Some of these microorganisms really don't like your high purity water. They seem to like those trace elements or minerals. So when you start transferring a process, one of the things that you may need to investigate when you hand your clone over or your strain over to another lab and they can't reproduce what you got, is start looking at some of the very basic things that are in your recipe and saying, are they truly equivalent? That could be a tough one if you think you're going to have to start shipping, your lab water to whoever you're manufacturing with. I thought maybe it was a fluke, but I've heard of this on multiple occasions, that something that simple can be a significant factor. And the same for a lot of the media components, especially if you get into things where you're using hydrolysates or peptones. There can be tremendous variability from batch to batch. So the more you go into more defined media, the better off you are. When we start looking at your primary recovery, there are things where when you do things on small scale on the bench, it's real easy. You just spin stuff down as fast as you can and you move on. As we move into manufacturing operations, you're going to have limitations based upon your equipment in terms of, if you're doing centrifugation for cell separation. Well, what volume are you going to be processing? What speed are you able to actually push it through your centrifuge? What you'll find is, as you scale up, you're going to have to start looking at process volumes, throughput, hold times, because that will be a real factor to consider. And so there are all those things that as we begin to work with you, I will start asking questions in terms of what do you see as your ultimate scale of product that you need, because that helps me to design the ultimate process that you will need based upon the volumes we have to handle, based upon how fragile the protein is and your expression system. Once you have your gamish of your either cell supernate or your lysed, cell paste and cell pellet, what you really want to do is to as effectively and efficiently capture your target molecule. So that's what your capture step is intended to do. That's why you see for antibody production, grab that protein A resin and use it because it is so effective. So that's something that what we want to do is to really pull that out and start getting rid of all of those host cell proteins, the DNAs, and those other components, and get it to the point where you have a protein that's relatively free and has, any proteases and other things that could interfere with your product away from it. So that's a trade-off. You've got expensive resins like protein A, but they're used, why? Because they're so effective and they can be reused. So that's something as you look at what you're manufacturing, you can use a novel resin, but you have to make sure that it pays off in the long run. Once we've done our development, we need to transition to the GMP manufacturing environment. If you're transferring a process to me, I'm actually going to ask you for not only your protocol, I'll ask you for what is the largest batch that you have manufactured. How many times have you done it? One time I had a client come to me. They were in a phase 3 study with a vaccine antigen. It was a conjugate antigen, and this was actually an extracted product. But they had, because they were in such a tight time constraint, they were essentially brute forcing the production of their antigen, where they had repeated this manufacturing process 99 times at a small scale because they didn't have the time to optimize the process, and they were constrained for purchasing new capital, so they couldn't effectively scale up. So we've seen that kind of thing, and we know it happens, and there are reasons for it. Ideally, though, what we would do is transfer in the process to our GMP manufacturing environment for an engineering run that's hopefully close to this intended scale of manufacture, or at least within tenfold. And the way we do it is we have our process development scientists work hand in hand with the GMP manufacturing personnel so that the GMP manufacturing personnel write the batch record with the review and input of the development scientist, recognizing that the manufacturing personnel are the ones that need to use the batch record, so they need to understand it. As I said, communication is everything, so you can see we've got arrows and lines going everywhere. And over all of this, we will have a project manager whose job it is to coordinate these activities internally and also externally with the customer to ensure that we keep everything on track and that details don't get lost or forgotten or slip out. What I'd like to do briefly is to share just kind of a little case study here where we worked with Pfenex to transfer a model protein from their site into our site in Jerusalem. And because we're a CMO, much of what we do is proprietary, so I can't share it unless we've made an agreement to do it as a press release. But at Pfenex, they developed the model protein. They were able to scale up to 20 liters fermenter, which is their largest capacity. They prepared a packet very similar to what I described in terms of, what is the protocol, what's the largest batch size, what are your parameters. And in this case, we actually did a paper document, essentially a paper transfer. Within SAFC, we took that, went up to a 75-liter scale, so about a little over, a 3 to 4-fold scale up. And what you can see is that we had very good comparability between what they achieved in their lab, from their progressive scale up from, the small scale mini fermentations up through the shake flask to the 20-liter, to what we were able to achieve in our 75-liter. So this just says, if you get the proper communication with appropriate documentation and a robust system, you can effectively do technology transfer. We did a lot of telephone calls and the like. I highly recommend that wherever possible, your scientists get together face to face to work through this, because no matter how well you think you write, there is going to be something that you miss when you put it on paper. I'd like to ask each of you, how many of you have gone to a party, potluck, had this absolutely fabulous dish, got the recipe from someone, go home and make it, and it doesn't taste the same? You've had it happen? Why not? What went wrong? You followed the recipe, how could you mess it up?
They left something out.
Well, that's the same thing. What happens? People don't put in certain steps that they just do automatically, or those, I'm notorious for it. My family would say that I experiment every night in the kitchen. I make tweaks constantly, as I'm cooking. Well, that's the thing that when you're writing your documentation does not get captured. So wherever possible, I say invest the time and money to get the scientists working side by side. It will pay off in the long run. So when we get down to it, as you transition from your discovery into your clinical material, we're going to need some things. Sheila hit well upon it that you're going to need specifications, not only for your API and your raw materials, but don't forget, you may have intermediates in your processing. You may need to put a step in your process where you have a defined hold time. It could be something like a cell paste. It could be something like a partially purified intermediate, where you may need to either be able to hold it at that step so you can do some further analytical testing. It may be you want to, in the future, be able to campaign material so you can make a larger downstream batch size. We talked about how all of this analytical testing eats material. Oh yeah, I can remember on one of the very first GMP runs we did, we thought we were making an astronomical amount of material. Guess what? By the time we did our QC testing, pulled stability and the retains, you're right, we had none left really to send to the client for what they needed. So that's something we actually look at now. How much material will I need for testing, retains, and stability? Because it's important. In response to, do we need to keep retains for the bulk drug substance? I highly recommend it. I just had a client where we actually needed to go back and do some additional testing and characterization on a product, and we're scrambling to get enough material from the stability and retains to do the additional testing because they wanted the regulatory agency asked for some updated analytical methods. So it is critical to keep that. As I said, we also, when you go through the key process parameters, that's something that we actually look at as we're developing specifications. We put the specs together looking at the ICH guidelines in terms of, the Q6B, and then we look at each of those and do we have steps in our process that actively control each of our critical quality parameters? And do we have enough information around that to know that at any given step in time, if we fall outside of the range of our parameters, we know what the impact will be? And it's one of those, I've been in the business long enough, you try to plan, cover every contingency, but guaranteed, there will be something that goes wrong. You know, if people say they have, 100% success rate, well, either they haven't been doing this very long, or they're not really being truthful with you. You know, we've got a pretty good success rate, but things can and will happen. It could be, humans are operating these plants, you have operator error. It could be, you know what, we had an equipment failure, it broke down, and now we need to know what to do about this. So things can and will happen, so the more you know about the process, the better off you are. And what we find as a CMO, we're also very actively involved in providing support for regulatory submissions, whether it's process information, characterization information, or the like. And we're seeing this more, that CMOs are being treated as true partners in activities and not just a vendor. So overall, when you're doing your transfers, what does success look like? It's having both parties truly understanding what your product is, what you need it to be, and being able to translate that into appropriate specifications. Wherever possible, we really want to get away from doing use testing. In my early days, I had a lot of products where I'd have to ship it back to the client because they would kind of need to do some type of use test because they hadn't formalized how to really put test methods in place to measure that particular characteristic. We need to understand enough about the process to make it robust so that we do have a high success rate when we manufacture. And it's very important that as you go through your tech transfer, whether it's internally from one team within your organization, from one lab member to another lab member, or to an outside CMO, that you learn how to effectively communicate, because in the long run, it is that communication that will ultimately make it a successful project or one where you struggle and have difficulties. And with that, I'd be happy to take any questions. Thank you.
On the list of things that you said that were needed for clinical development, I noticed that you had indicated analytical method development. Are you proposing that even for phase 1, you have to have those methods validated?
Oh, no, not valid. Okay, that's something. Yeah, when we are developing products, we actually have a matrix that we use for what needs to be put in place at which stage of the clinical program. So we do need methods developed for phase 1 with some level of qualification, and we will actually look at the methods and determine, essentially which are critical quality attributes, or which methods apply to those quality attributes, those we want to have in their more mature state. For stability indicating assays, we do prefer to have those validated because that data will be carried out for multiple years. Then as we move from phase 1 into phase 2, that's when we begin doing our analytical method validation. Some of this will be dependent upon client requirements or other regulatory requirements. We've had experience where some agencies do tend to, or some locations do tend to validate earlier than others. But yeah, no, we do not, for early phase work, we do not put full validation in place. Very often the product is not well enough defined, and the analytical methods are usually not fully mature at that point. There's not enough lot history to do that. But good point, yeah.
Well, just to follow up, did you indicate that you do try to have your stability indicating methods validated early? The problem that I see with that is the process is not yet locked down.
Right.
And so you, in fact, may think that you have a stability indicating method that is not suitable for demonstrating stability because there exists some material that you didn't know existed, because the classic is if you don't have the test, you can't know that it exists. But
Right, yeah, and that's always a question, because you also get to the point where, you don't want to just begin randomly testing for things, because what do you do with data that you aren't quite sure what it means? Once you collect it, you do have to do something with it. So it's that fine balance between having your product well characterized, but then having data that it may not be fully relevant. Okay, thank you. Yeah, Keith.
Oh, very nice presentation, thank you. Quick question on batch-to-batch variability and the end amount of material you may need may actually require some kind of lot combinations to blend different lots together. What are some of the challenges that you're seeing in GMP manufacturing is trying to actually address those things early on,
Well, one of the things that we see in terms of batch-to-batch variability, that's why we always recommend, where possible, to have more than one lot of material manufactured, especially for clinical material. We recognize very often that's difficult, but you'll see in the early stage work what we said is, 2 to 3 batches. What we want to do is to establish what range of variability do we expect to see in a process. In terms of blending, you can't blend material unless it all meets specification, but what we see is that for early phase material, specifications are generally set more broad, and then as you progress into your later stage clinical material, you begin to tighten those as you both understand what your product needs to be and you understand how to better control your process. But it's something that we have seen where it is challenging for some programs where there's limited resources, or the batches of material are large enough to support both phase 1 and phase 2 studies. Having adequate lot history available to truly understand what acceptable ranges are for variability. So that's something where it's going to be that balance between, I would almost recommend, if you're able to, is to trade off and do some multiple smaller batches to be able to get adequate lot history than one large batch that carries you for multiple phases of your clinical study. You really don't want to be in the position where you've done one magic lot and find out that's truly a magic lot. Okay, next.
You mentioned early on about product changeover, cleaning, obviously.
Yep.
The only time I've ever grown bacteria and yeast is when I've made a mistake. So I don't have experience in this area. Is it single use? You're not using it because there's not enough agitation or
We
oxygen?
use single use or disposables where appropriate. For fermentation, at least for microbial systems, we have not found that it seems to work as well, at least in our hands, for the scales at which we're doing it. It's much more prevalent in mammalian cell culture, and I know in our facility in Carlsbad, California, where we do viral vectors, we routinely use disposables, but the wave technology seems to work much better there.
Do you think it's agitation?
Yeah, I think, agitation or oxygen content, but we think it was more just due to that or the geometry that's required. If someone has experience with disposables for microbial fermentation that they have found very positive, then I'd be more than happy to talk with them about that. We do tend to use a lot of disposable or bag technology for once you get your product partially purified, in terms of, for collecting or storing or transferring materials. They're wonderful. We're seeing more and more utility for, ultrafiltration cassettes where things are, almost single use. It's always that trade-off between cost of purchasing membranes and cost of verifying, cleaning, and reuse. The same for chromatography columns, those are now becoming available. So I agree, it eliminates a lot of the cost for the clean verification studies and some of the changeover.
I'd like to follow up on that. It may eliminate a lot of the changeover issues, but it introduces issues of leachables and extractables. So you need to weigh that. Additionally, there are cell lines which work very well in the disposable from company A, but actually do not work well with the disposable from company B, even though they are, quote, the same plastic. So it is often presented as much simpler than the reality.
Sorry.
Yes? Oh.
You were talking about defining critical process parameters very early on. By that, do you mean just figuring out which process parameters are critical, or actually defining the range for the small-scale process, and there's some way of figuring out what they will be when you go to a larger scale?
Generally what we do is try to identify which ones we think will be critical parameters, because what we find is that we like to get at least some design work in to get, what are our suitable operating ranges. But it truly is not until you get into some of your larger scale work where you can verify that it's appropriate. You need to be able to understand what your effective scale down model is. And that's what, as you move forward, that's what I would use for my true range finding studies. But you need to do some at least initial work to understand when you go into manufacturing, what is the window under which I can operate. But I think it's important to, early on in your product development, to have a good understanding of, which test methods do you think are going to be essential and critical in terms of monitoring your ability to make the product robustly. And I agree, there are things that we find that come out after the fact that, gee, I didn't think that would be critical, but it sure turned out to be. I can think of one where we found out that, like the acetate content turned out to be critical because it turned out to help control pH. And we thought, oh, well, it's just going to be sort of a byproduct of how the process is carried out and how the lyophilization is performed. But we found out that it could be critical, and that's something that initially we didn't anticipate that. So it's a balance, especially as you move forward. It used to be we'd just, get a product to work, run it multiple times, and then say, oh, okay, if we do the statistics, do a t-test, add our 2 standard deviation units, well, this must be what the spec range is. Now we need a little bit more thought behind it. But yeah, it's one of those balances. But when you're going through your early development, you can't test every single parameter that you may think is necessary early on as you're doing your study. So, we try to identify what we view as the key ones. Yes?
Again, just if you could speak to the transition. You mentioned a little bit on the media components and some of the raw materials. It's like, I can see there's a big trouble with going from bench scale in, say, San Diego or Boston to trying to go to GMP production in the Middle East, where you're going to have some raw material challenges there. I was wondering what your experiences are on that, might be able to think about some of those things early on in development.
Yeah, and that's why I say in many respects, the earlier we get involved, the easier it is to transition. One of the things that we do as a corporation is we actually do qualify vendors for our raw materials. So if we're working with you, we're going to tell you who the vendors are that we've qualified. I can tell you there are some media component vendors that we have chosen not to qualify because we don't feel they produce a reliable, consistent product, or they cannot provide the proper documentation. So if you have developed a product, what we will try to do is if the vendor and the components are suitable, we will use those. When we go into manufacturing, we do try to use compendial grade reagents wherever possible. We, do have, the peptones and that, which, where possible, we also try to use non-animal derived materials to avoid those concerns. And then we ensure that we know the vendors, and before going into GMP manufacturing, we will at least have a paper audit of our vendors to qualify them. So we have a matrix in terms of how we determine which raw material vendors, go through kind of a paper audit, which ones go through on-site audits and the like, and that'll be based upon, the criticality of the raw material, the volume used and that kind of thing. But that's something where we will try to work with you. And one of the early things we would do if we need to change raw materials between what you've used in bench and what we have available is to do that small scale batch, preferably at the scale that you've successfully performed, and demonstrate that we get comparable results. And that's something, where we recognize you're more likely to have batch to batch variability, and that's where, it comes into, if you can get multiple lots and verify that, yep, more than one lot works, that's the preferred way to do it. Yes?
Would you comment on that, trend we see like in small molecules, number of GP services coming from Asia, India, et cetera, given all the complexities, it's like what we see happening in China is a threat just to, strong force in the United States, or because clearly people claiming price advantages, but would it be like sacrifices the quality, or what's your opinion on that?
Well, and I think that comes down to knowing who you're working with. I do know that we've had many clients that have expressed concerns about operating in, China or some of those areas due to the concern over quality and maybe less access to facilities to be able to do the audits. So, that comes back to generally it's true if you're operating in, the US or Europe, or like we do in Israel. In general, you are going to have a higher cost, labor cost associated with it. So that's where you have to understand what are the quality requirements of the company that you're working with. Can you verify that they operate under the guidelines that they say they operate? And that's where if you don't feel you have the transparency that you need working with our quality and regulatory personnel, then I would say that's where you need to be very cautious about where you're going to achieve an effective cost of goods. It's going to be a lot less expensive for you in the long run to get a high quality product that's made correctly the first time than to go through a series of recalls. Any other questions? Okay, thank you.